My name is Dr. Rhonda Voskuhl. I am the Jack Skirball Chair, the Director of the UCLA program, and a professor in the Department of Neurology at UCLA. A few days ago, I just finished being president of the Organization for the Study of Sex Differences. So today I'm going to speak to you about using sex differences to address an unmet need for a treatment targeting cognitive deficits at menopause. These are my disclosures. So I'm going to go to the endpoint in the beginning, and that is that I'm going to propose that sex differences are due to sex chromosomes that occur across the lifespan, but overlaid on top of that, there's an effect of sex hormones. And these sex hormones can have differential effects depending on when they decline during aging. For example, testosterone in males stays pretty high until age 30 or so, and then it drops very, very gradually to about the age of 80 in men, andropause in men. In contrast, for women, estrogens stay quite high until about age 50, and then there's an abrupt drop between 50 and 55 years old, and then there are very low levels thereafter. I'm going to propose that both estrogen and testosterone are neuroprotective, and that loss in either one is deleterious. And then you can see that at this particular part of the lifetime, less than 50 years old, for example, males could actually be doing worse with neurodegeneration. In contrast to at this part of the lifetime, 60 years or older, women could be doing worse. So let's see if there's any evidence to that effect. Well, as this group is well aware of, aging is extremely important during health. It's associated with cognitive decline, brain atrophy, and neurodegeneration. Indeed, age is the most important risk factor for neurodegenerative diseases. Now, some aspects of aging are not reversible likely, but some others may be reversible or preventable, such as the loss of hormones with menopause or andropause. So what happens in aging during multiple sclerosis? It's actually really interesting. It goes at a young age, it's a very inflammatory disease, and at a late age, it's a neurodegenerative disease. So instead of having relapses, they have worse disability. Instead of having a lot of enhancing lesions on MRI, they have brain atrophy. It's also been shown in MS that loss of estrogen and menopause and loss of testosterone and andropause both appear to be worse for exacerbating pre-existing disabilities. What about aging during MCI and Alzheimer's disease? Well, it's thought also here again that loss of sex hormones is probably bad to exacerbate both of these conditions, whether it's andropause or menopause. Now, I'm not surprised that there could be a similarity in effects of these two different sex hormones in the sense that testosterone is converted to estrogen by aromatase in the brain. And so in either case, you would have less estrogen receptor ligation. Why is this important? Well, it's important because estrogen receptors are expressed on neurons, oligodendrocytes, microglia, and astrocytes. Here they are. Your alpha and beta expressed on these critical cells that would be involved in neurodegeneration. And of course, during the menstrual cycle, estradiol binds these. Estradiol binds alpha with a high affinity, also binds beta, but it can have deleterious effects on breast and other situations. The estriol is kind of interesting. It's a pregnancy estrogen, and it's thought to be weak, but actually what it does, it binds to ER beta well over ER alpha, and that's why it's thought to be a safer estrogen. Now, of course, testosterone, when it's converted to estrogen, is converted to estradiol in men by aromatase, as I said, in the brain. So I think this shows how ligation of estrogen receptors in brain could have a major role in neuroprotection. Now let's talk more about these sex differences with aging. Well, when you think about sex differences in the brain with aging, you first have to think that, well, there are sex differences in the brain all the time, during development, during the young adult life, and during aging. So sex is a biologic variable, not just in autoimmunity or the heart. It's sex differences in the brain have been shown for decades. And notably, this isn't all due to, it could be in part due to social or environmental conditions, but actually sex differences in the brain occur across species from humans to mice. So this suggests that there are clearly some basic biological differences in sex hormones and sex chromosomes effect on the brain. Now, these sex differences have been shown at the cellular and molecular levels. You can read about that in some of these references I have. In addition, it's known that males' brains are larger than females'. Male brain size is proportional to skull size, which is proportional to body size. And so, for example, Shaquille O'Neal's brain also will be bigger than Albert Einstein's. Men are bigger than women are. That said, what's very interesting is there are sex differences in brain substructures after correcting for brain size. So, such as within the hippocampus, dorsal and ventral hippocampus are quite different. One's larger in men and one's larger in women. And what about sex differences, not during health, but during mild cognitive impairment and Alzheimer's disease? Well, it's known that 2/3 of Alzheimer's disease patients are women, and this is not merely explained by longevity. There are many groups that are studying this, and rightly so. It's a very important problem. What I'm interested in the current issue is that women actually have a higher rate of progression from MCI to AD at ages greater than 60, and they have a faster rate of brain atrophy as compared to men. But what's the contrast to this is that at relatively younger ages, less than 60 years old, men have a higher rate of progression from MCI to AD. And also it may be that men are more risk for MCI. That's somewhat controversial, but it's certainly not the case that women are more at risk for MCI at early ages. So this is really interesting, this effect of age on MCI and AD risk. So what about MS? Well, of course, MS affects women more than men, but it's interesting to note when you talk about neurodegeneration, when men get MS, they do worse with respect to their disability progression. They've been shown to have worse cognitive disability and worse gray matter atrophy. Keep in mind that when the MS studies are done, they're done in patients that are 18 to 65 years old with a mean of about 40 years old. We studied this in some detail. It's kind of interesting to see. Our group at UCLA worked with the Charité Hospital in Berlin. There were 134 patients, 89 MS and 45 age and sex matched healthy controls. Again, mean age of 40 years. What we did is we didn't compare MS women to MS men for substructure brain atrophy. Instead, what we did is we compared MS women to healthy women and MS men to healthy men. The reason we do that is because otherwise, you wouldn't know if it was due to just sex differences in substructures during health. We wanted to know what was the effect of disease in women, what was the effect of disease in men. And when we did that, you can see that these women have thalamic atrophy. It's known to occur. It's relatively early, as you can see. Men also had thalamic atrophy, but they also had this spreading to the cerebral cortex and other regions. This was done by Voskuhl-based morphometry. It was also validated using free surfer, looking at cortical thinning, which occurred in males but not females. So this is consistent with, at this early age, mean age of 40, with males actually doing worse with NMS, with regional brain atrophy. Well, the question then, of course, is, well, what happens here when women lose estrogen so abruptly and dramatically during menopause? Well, menopause NMS has been studied for a while, but not in a lot of depth. And originally it was known for decades that women reported that their pre-existing symptoms got worse with menopause. Fortunately, objective data began to emerge where people were examined with this EDSS disability scoring composite every year, and people were getting MRIs at universities every year. And by and large, this showed that there was an inflection point whereby women's disabilities were getting worse at menopause. And one group actually showed that the menopausal marker, anti-Mullerian hormone, lower amounts of that were indicating early menopause did indeed correlate with not only disability worsening, but also lower cortical gray matter volumes. There was cortical atrophy that was also occurring in menopause. So now we have objective data on exams and now MRI showing that women with MS do worse during menopause. What about menopause, its effect during healthy women? Well, that's widely known that women during health have cognitive difficulties. It's called brain fog. It's got a layman's term. It's so widely known. And this has been quantified objectively by neuropsychologists using domain-specific tests. What menopause does to women is it impairs verbal memory and processing speed. So it's not global cognition. It's not IQ. It's none of those things. What it is, it's very specific cognitive domains that are affected by menopause. MRIs, of course, have also been done, and it's been shown that both surgical and natural menopause cause problems with atrophy in the hippocampus and prefrontal cortex. They also have shown that you can have functional connectivity abnormalities in the hippocampus and prefrontal cortex during natural menopause. Also, the ApoE4 carriers that are healthy, and also those with the MCI and MS have shown hippocampal CA1 atrophy and thinning of the entorhinal cortex. So this is all pointing to a very cognitive domain-specific and brain region-specific problem. So let's move on to the final phase. And this is, well, how can we fix this? How can we prevent this abrupt drop in estrogen with menopause? How can we treat it? And that's been extremely hard. We thought we'd go back to the drawing board to see if we could take a a cell-specific and region-specific approach. So what we did, this is a paper that's in revision currently. We took mice that were aging, they were young, which is 3 to 4 months, midlife, which is 12 to 14 months, and old, which is 21 to 22 months of age. Red is female, blue is male. What we showed was that when we do, these mice do the Morse water maze, they get trained and then they go to the target quadrant over the other quadrant. Statistically, they should, because they've learned that. And this really is pretty much intact. This is between groups comparison. This is of just the target quadrant. This is target compared to other. You can see in the red that they actually significantly recognize the target. Older females look a little bit worse, but they still are doing okay. They're not significantly worse. Males are not having a problem. They're all the way through age. They're doing fine on this behavioral test. But then we said, well, usually MRIs are subclinical measures that are more sensitive to behavioral than the behavioral test. And so we did MRIs on these same mice, and we showed that something very interesting. The males were having, in blue, were having a gradual loss from young to midlife to old of frontal cortex volume and striatum volume. In contrast, the females were maintaining their volumes pretty well through midlife. They had no problem, but then after midlife, you had this abrupt drop at old age. Same thing here, maintaining volumes in red and the abrupt drop at old age. Now, dorsal hippocampus never actually atrophied in the males, but females again showed maintenance from young to midlife, but then this drop at old age. So this was very intriguing. The next thing that we did is we said, well, I wonder what would happen if we take these midlife females. The key thing here is midlife. And we said, well, what would happen if we gonadectomize them? So they abruptly lost their estrogen. So when mice, they were all actually gonadectomized at 2 months of age, but then they were followed and tested at either 3 to 4 months of age in the young or at 12 to 14 months in the midlife. What we found was, this is the target quadrant. You can see that when you gonadectomize the mice and examine them in young adulthood, it really doesn't cause a significant problem with the behavioral test. However, when you do this, when you examine them at midlife, it does. So what this means is that, and this is another cognitive test, which I won't go into, same thing. They're fine when they're young and they get gonadectomized, overectomized, but when they are examined at midlife later on, they're having difficulties. What this means is there's a very interesting and important age by sex hormone interaction. It's not all about sex hormones. It's not all about age, but when you get the aging and the loss of sex hormones together, that's what's really bad. We then did, of course, the dorsal hippocampal atrophy, and we asked whether we could make the midlife ones look as bad as the old ones, because the old ones had volume loss, and sure enough, these are the old ones that are sham and GDX. When you look at the midlife ones, this is the sham, they're gonadally intact, versus the GDX. You see the GDX is bringing them down to the atrophy level of the old ones when they're still midlife, but they're GDX. We then aligned the dorsal hippocampal volume with the time and target quadrant to show that our imaging was relevant to our clinical outcome. Then we said, well, I wonder what the pathology is in the dorsal hippocampus at midlife when they're GDX or not, right? That's really interesting. So we stained, here's an example, PSD95 is a synaptic stain in red, GFAP staining astrocytes. And you can see in the sham compared to the GDX, in the GDX, they have more astrocyte activation than they have synaptic loss. This is all quantified for astrocyte activation, microglial activation, and synaptic loss here. And you can see from young to midlife, again, not a lot of problems when you GDX the young ones, but when you GDX, they get older, it is kind of a problem, but when you GDX them, you really exacerbate things at midlife. So again, once again, it's an age by loss of sex hormone interaction on pathology as well. And here's the synaptic loss that's quantified, with the worst always being the midlife GDX. We then quantified the synaptic stain, PSD95, with time and target quadrant to put together our neuropathology with our behavioral test. So then we said, well, I wonder, let's do something more surgical than just, or more precise, I should say, than gonadectomy. Let's go in and try to figure out what cell and what receptor is mediating this protection and what causes the loss of it. So we actually focused on astrocytes, because astrocytes can not only activate microglia, they actually maintain the synaptic integrity. Again, astrocytes do a lot of things, but this is really interesting in the context of what we were thinking about. So what we did is we did, in this set of experiments I'm going to show you, we did not gonadectomize them. What we did is we left them gonadally intact. So they had estrogen circulating, but what we did is we knocked out estrogen receptor beta either in astrocytes or neurons. So either in astrocytes or neurons, we knocked out ER beta to see what would happen. Could we recapitulate this gonadectomy picture that we had just seen? And what happened was, this is the cognitive testing and this is the MRI. What happened was, this is the wild type. These are all midlife females. Wild type, this is ER beta knocked out in astrocytes. This is ER beta knocked out in neurons. And what you see is when you knock an ER beta out of astrocytes, they have a significant impairment in recognizing the target quadrant over the opposite quadrant. This is also true in another test that I'm not going to get into, but interestingly, it didn't matter if you knocked an ER beta out of neurons, it didn't have any functional significance. So these receptors can exist on several cells, but you have to go in vivo and knock it out in a particular cell to determine the mechanism of functionality. This is hippocampus, dorsal hippocampus and ventral hippocampus. And again, this is what we've seen over and over. Here with the knockouts, though, showing that dorsal hippocampus is affected. Ventral hippocampus is not affected with gonadectomy, is not affected with the knockout. And then whole hippocampus is a reflection of dorsal plus ventral. And looking into the detail, again, as we showed with the behavioral testing, when you knock ER beta out of astrocytes, you get this dorsal hippocampal atrophy, but you don't get it when you knock ER beta out of neurons. So I hope this shows you that we can start thinking about how to treat this abrupt loss of estrogen with menopause, and the target, a good target would be ER beta in astrocytes and dorsal hippocampus. So when we think about treating menopausal women, healthy menopausal women, it's been done on and on, but what's been done are just repurposing treatments that are used for non-cognitive problems. And so that's not the answer. We need to actually leverage off of what's been learned, and that is the estrogen timing is important. You have to treat within 5 to 10 years of menopause, most likely, clearly before the age of 65. The estrogen type is very important. Estradiol, there's data showing that it may work and cause region-specific and protection, but Premarin doesn't. It would actually be a negative control, in my opinion, for neuroprotection. The estrogen dose is critical. Higher doses are better, but again, with estradiol, you can only push the dose so high because it binds to ER beta. So we have to use another selective ligand that doesn't bind to ER alpha. I would suggest ER beta. The benefits that have been observed previously, again, it's hard to review all this, but there have been benefits with estradiol at a given dose on hippocampus and frontal cortex, atrophy and other measures on MRI. No effect on global cognition, but you wouldn't expect it. It should be testing specific domains. So this is kind of where we're at. We basically just repurposed drugs that have been used for non-cognitive symptoms and hope they were neuroprotective. And I think we need to do better than that. Is there anything we can learn from MS? And there actually is, because in MS, now these were not menopausal women, they were younger, they were about age 40. And what was done there were 2 trials showed some promising results. So an Italian group gave ethinyl estradiol, which binds ER alpha more than ER beta. And what they showed in a low dose versus high dose birth control regimen, they showed that fewer patients had cognitive impairment on aroused brief repeatable battery at the higher dose, but not the lower dose of estradiol. So that's interesting. There was some promise. Our group actually did a trial that showed that women were treated. We were treated with estriol, not estradiol, because we like estriol since it binds ER beta over ER alpha. Therefore, it would be safer. And we gave a pregnancy dose actually, because we could, because ER beta is safer than ER alpha, and pregnancy is good for MS. Bottom line is we found that there was an improvement in cognitive testing, specifically processing speed in the estriol group compared to the placebo group. And notably, the cognitive testing improvement, the level of improvement in cognitive testing correlated with the estrogen blood levels. So higher estrogen blood levels, the more cognitive improvement they had. In addition, we showed that estriol caused reduction in cerebral cortex atrophy. And furthermore, the level of cerebral cortex atrophy reduction correlated with the cognitive testing improvement. So altogether, the MS field has told us that you can get cognitive improvement in MS, and specifically its processing speed, if you use an estrogen dose that's high enough and you get an estriol blood level that's maintained, and also you're targeting and looking as your outcome measure things like specific, again, domains like processing speed, and here on MRI, cerebral cortex. This is data that is in revision that I wanted to show. That's pretty interesting because we measure serum neurofilament levels because they've been used as a marker in aging and MS for neurodegeneration. This is our serum NFL data in estriol-treated MS patients in red compared to placebo in blue. And as you see, the people in blue did not have, the subjects in blue, the MS patients did not have a reduction in NFL, whereas the ones on estriol did at month 12. So this is very exciting to see a reduction in NFL at month 12 in the estriol treated patients. So to start to wrap up, I think we should, mind the gap. There's a huge gap in knowledge and particularly treatments for women targeting cognitive deficits. We need to, and again, this affects 50% of the population. At some point in their life, women will have or undergo or be concerned about menopause and their cognitive difficulties. So we need to go beyond merely repurposing HRT for non-cognitive symptoms. We need to actually design and optimize the treatment targeting and hoping to achieve neuroprotection. We can, however, use the extremely valuable past insights of estrogen dosing, which is clearly dependent on timing, the age, the type of estrogen, and the dose of estrogen. So we can learn a lot from those things, but we do need to move forward with a specific approach designed for neuroprotection and to achieve region-specific, cell-specific, and I would say even hormone receptor-specific ligation in order to have a new drug to treat cognitive decline in menopause. So finishing up the trial design, I would say there should be a call to action to prospectively design clinical trials for estriol or beta ligand treatment in menopausal women. The outcomes, just to be clear, should not be global brain atrophy improvement. They should not be global cognition. What they should be is improvements on regional brain atrophy, that'd be frontal cortex and hippocampus, and it would be cognitive domain-specific testing, processing speed, and verbal memory. So those should be the primary and secondary outcome measures, not these more global measures. I want to thank the UCLA team that's just been incredible in this work, particularly the two grad students who now became PhDs and are off in their dream jobs, Cassandra Meyer and Lisa Golden. These are our graduate students and undergrads in the lab that are just so critical to what we do. And then the faculty, Noriko Ito, Alan McKenzie Graham, and Yuichiro Ito in the program, as well as the other clinicians and collaborators with the UCLA MS program. And these are our funding agencies, which I always want to thank very much. So I will go ahead and end there and take questions. Thank you.